Today, this is the Professor, CBMD PhD. It's Thursday, June 1st, 2023. It is 3:45 and 25 seconds, a.m. Eastern Daylight Time. And today's research, Unveiling Harate. So let's jump to it here. The scientists unveil Harate, guided mechanisms, driving cell fate. Date May 30th, 2023. Source, Monash University. The early stages of embryonic development contain many of life's mysteries. Unlocking these mysteries can help us better understand early development and birth defects and help develop new, regenerative medicine treatments. Researchers have now characterized a critical time in mammalian embryonic development using powerful and innovative imaging techniques. As just a few days into the journey of embryogenesis, when turning into 16 cells, the embryo must make its first difficult decision: which of the cells will give rise to the embryo and will become extra embryonic tissue, for example, placenta. In this study, the research team has discovered how the decision-making process is facilitated by capturing the inner organization of single cells of the early embryo. Ribonucleic acid, RNA, plays a key role here. At the 16-cell stage, the different subtypes of RNA, named rRNAs, mRNAs, and tRNAs, are sorted to the two ends of a cell called apical and basal side. The distribution of RNA subtypes determines what the next generation of cells of the embryo will become. Interestingly, while most mRNAs and tRNAs remain part at the apical side, most rRNA molecules travel down to the basal side, hitchhiking on organelles called lysosomes. Even though retaining less overall RNA content, the apical sides of outer 16-cell stage cells contains a full collection of RNAs and other factors required for protein production. The crowded basal side, however, is occupied predominantly with rRNAs, daughter cells obtaining the more active protein factors to the apical side are more transformable and specialized into the future placenta. The daughter cells, which retain the potential to still become any type of cell of the adult organism called pluripotency, receive the less translationally active bulk of rRNA. And this decision, and many like it, which is known as cell fate, are important in development as it determines how these early cells reach their final cell type, such as skin cells, heart muscle cells, and brain cells. As for regenerative medicine, being able to orchestrate cell fate opens up the capacity to generate new stem cell-based treatments for a number of diseases and conditions. As in real life, cells can influence the direction of their own future by getting organized early. The research may open new ways to predict and direct cell fate decisions. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.